Walking on two legs defines us as human beings more than any other physical characteristic. This seemingly simple ability required one of the most complex evolutionary transformations in the history of life on Earth. Every step you take represents millions of years of precise anatomical engineering that distinguishes our species from every other primate that has ever lived. The human pelvis tells this extraordinary story. Unlike any other vertebrate structure, our pelvic anatomy underwent radical modifications that enabled upright locomotion while simultaneously solving the challenge of birthing large-brained infants. This transformation began approximately 5 million years ago and involved changes so fundamental that they affected every aspect of human biology, from our skeletal architecture to our reproductive physiology. Recent scientific breakthroughs have finally revealed the developmental mechanisms that created our unique anatomy. Through meticulous study of embryonic specimens from humans and other primates, researchers have identified two critical evolutionary innovations that occurred at precise moments in our developmental timeline. These discoveries explain how a creature designed for life in the trees evolved into the only primate capable of efficient bipedal locomotion. The implications extend far beyond anatomy. Standing upright freed our hands for tool manufacture and complex manipulation. It elevated our visual perspective, enhancing our ability to scan environments and communicate across distances. The energy efficiency of bipedal locomotion enabled long-distance migration and persistence hunting strategies that would prove crucial to human survival and expansion across the globe. Understanding this transformation requires examining evidence from multiple scientific disciplines. Paleontologists have uncovered fossil footprints and skeletal remains that document the timeline of bipedal evolution. Developmental biologists have traced the embryonic processes that create our distinctive pelvic structure. Geneticists have identified the molecular mechanisms that orchestrate these developmental changes. Together, these findings provide the most complete picture yet of how humans became upright. The story begins with a fundamental biological mystery that has puzzled scientists for over a century. All vertebrates possess a pelvis, but only humans evolved one specifically adapted for upright walking. The familiar short, wide shape of our pelvic bones differs dramatically from the tall, narrow configuration found in our closest living relatives. This anatomical distinction enables us to balance efficiently on two legs while maintaining the structural support necessary for our internal organs and reproductive system. The transformation required solving multiple engineering challenges simultaneously. The pelvis had to provide stable attachment points for the powerful gluteal muscles that maintain balance during bipedal locomotion. It needed to position the hip joints directly beneath the body's center of gravity. The structure had to accommodate the distinctive S-shaped curvature of the human spine that distributes weight efficiently during upright posture. Additionally, the pelvis had to maintain adequate space within the birth canal to allow passage of our species' characteristically large-brained infants. For decades, scientists could observe the end result of this evolutionary process in fossil specimens, but the underlying mechanisms remained mysterious. How did natural selection modify the developmental processes that build the pelvis? What genetic changes enabled such dramatic anatomical restructuring? When did these modifications occur during human evolutionary history? The answers lay hidden in the earliest stages of human development, encoded in the precise timing and orientation of bone formation during embryonic growth. By comparing human embryonic development with that of other primates, researchers finally identified the specific developmental innovations that enabled our upright posture. These discoveries represent one of the most significant advances in understanding human evolution in recent decades. The quest to understand human bipedalism begins with some of the most compelling evidence in the fossil record. In the volcanic ash beds of Laetoli, Tanzania, paleontologists discovered footprints that have captivated scientists and the public alike for nearly 5 decades. These tracks, preserved 3.6 million years ago, provide direct evidence that our ancestors had already mastered upright walking by the mid-Pliocene epoch. The Laetoli footprints reveal extraordinary details about early hominin locomotion. Analysis of the track patterns demonstrates that Australopithecus afarensis individuals walked with a heel-to-toe gait virtually identical to modern humans. The footprints show evidence of a well-developed longitudinal arch, a crucial adaptation for efficient bipedal locomotion that helps absorb impact forces and provide spring-like energy return during walking. The spacing and depth variations of individual tracks indicate these early humans possessed the same basic biomechanical patterns we use today. Complementing the footprint evidence, the skeleton of Lucy, discovered in Ethiopia's Afar Depression, provides detailed anatomical proof of bipedal adaptation. This nearly complete Australopithecus afarensis specimen, dating to 3.2 million years ago, preserves crucial elements of the locomotor apparatus. Lucy's pelvic bones display clear modifications for upright walking, including a shortened and broadened ilium that provided enhanced muscle attachment sites for maintaining balance during bipedal locomotion. The femoral anatomy in Lucy's skeleton reveals additional adaptations for bipedalism. The femur displays an angled orientation that positions the knee joints directly beneath the body's center of gravity, a configuration essential for efficient upright walking. The greater trochanter, a bony prominence where major hip muscles attach, shows enlargement consistent with the enhanced muscular requirements of bipedal locomotion. These modifications represent fundamental departures from the anatomy of quadrupedal primates. Examination of Lucy's vertebral column provides evidence for the distinctive spinal curvature associated with upright posture. While the preservation of vertebrae in early hominin fossils is often incomplete, available evidence suggests the development of increased lumbar lordosis, the inward curvature of the lower spine that helps position the torso over the legs during bipedal stance. This spinal configuration works in conjunction with pelvic modifications to create an integrated system for upright locomotion. Additional fossil discoveries from sites across Africa have expanded our understanding of early bipedal evolution. The partial skeleton of Ardipithecus ramidus, dating to 4.4 million years ago, provides evidence that bipedal adaptations were already present in some of our earliest hominin ancestors. While Ardipithecus retained many features associated with arboreal locomotion, the pelvic anatomy shows clear modifications toward bipedal capability, suggesting a mixed locomotor repertoire during the early phases of hominin evolution. The fossil record documents a gradual refinement of bipedal adaptations over millions of years. Later specimens, including those attributed to Homo erectus and archaic Homo sapiens, show continued evolution toward the fully modern pattern of pelvic anatomy and lower limb morphology. This progression demonstrates that bipedal locomotion underwent continuous optimization throughout human evolutionary history, rather than appearing as a single, sudden transformation. However, the fossil evidence alone cannot explain the developmental mechanisms that enabled these anatomical changes. Bones and footprints preserve the results of evolutionary processes, but provide limited insight into the genetic and developmental innovations that made such modifications possible. To understand how evolution actually remodeled the hominin pelvis, scientists needed to examine the processes that build these structures during individual development. The limitations of the fossil record became particularly apparent when researchers attempted to understand the specific sequence of anatomical changes that occurred during the evolution of bipedalism. While fossils could document the end states of evolutionary transformation, they could not reveal the intermediate developmental stages that connected ancestral and derived conditions. This gap in understanding necessitated a different research approach, one that would examine the actual processes of bone and cartilage formation in living primates. The transition from fossil evidence to developmental analysis represents a crucial shift in methodology for understanding human evolution. Rather than relying solely on comparative anatomy of preserved remains, researchers began investigating the dynamic processes that create anatomical structures during embryonic and fetal development. This approach promised to reveal the actual mechanisms by which evolution modified our species' distinctive anatomy. Before examining the developmental processes that create our unique anatomy, we must understand the profound structural differences between human and ape pelvic architecture. These anatomical distinctions represent the end products of millions of years of evolutionary divergence and provide the foundation for understanding how our species acquired the ability to walk upright. The chimpanzee pelvis exemplifies the ancestral primate condition from which human anatomy evolved. In chimpanzees and other great apes, the iliac blade extends vertically along the spine, creating a tall, narrow configuration well suited for quadrupedal locomotion and arboreal movement. This elongated structure provides extensive attachment surfaces for the large dorsal muscles that power climbing and brachiating behaviors. The narrow width of the ape pelvis reflects the mechanical requirements of a body plan designed for movement through three-dimensional arboreal environments. The orientation of the chimpanzee iliac blade positions the major hip muscles to function most efficiently during quadrupedal movement. The gluteal muscles, which in humans serve primarily to maintain lateral balance during bipedal walking, function in apes as powerful hip extensors during quadrupedal locomotion. The vertical orientation of the ilium provides optimal mechanical advantage for these muscles when the hip joint operates in the extended position characteristic of knuckle walking and climbing. Human pelvic anatomy presents a dramatically different configuration. Our iliac blades are short and broad, extending laterally rather than vertically to create the characteristic bowl-shaped pelvis that supports upright posture. This fundamental reorientation positions the hip joints directly beneath the body's center of gravity, a requirement for efficient bipedal balance. The lateral expansion of the iliac blades provides attachment sites for muscles that function as lateral stabilizers during single-leg stance phases of walking. The functional significance of this anatomical transformation becomes apparent when examining the biomechanics of bipedal locomotion. During each step of human walking, the body must balance on one leg while the opposite leg swings forward. The broad configuration of our pelvis provides attachment sites for the gluteus medius and gluteus minimus muscles, which contract to prevent the pelvis from tilting toward the unsupported side. This lateral stabilization mechanism is essential for maintaining balance during bipedal gait and represents one of the most important functional adaptations for upright walking. The human pelvis also accommodates the unique requirements of our species' reproductive biology. The broad, bowl-shaped configuration maintains adequate space within the birth canal to allow passage of large-brained infants. This accommodation represents a crucial evolutionary compromise, as the optimal pelvic shape for locomotor efficiency differs from the optimal shape for safe childbirth. The human pelvis represents an evolutionary solution that balances these competing demands. Detailed analysis of pelvic morphology reveals additional adaptations for bipedal locomotion. The human sacrum, The fused vertebrae that form the posterior wall of the pelvis displays increased breadth and altered curvature compared to apes. These modifications help distribute weight forces efficiently during upright posture and provide enhanced stability for the vertebral column. The sacroiliac joints, which connect the sacrum to the iliac bones, show modifications that accommodate the increased forces generated during bipedal locomotion. The acetabulum, or hip socket, displays species-specific modifications that reflect different locomotor demands. In humans, the acetabulum faces laterally and slightly downward, positioning the femoral head to support body weight during upright stance. The depth and orientation of the hip socket also influence the range of motion available at the hip joint, with human anatomy optimized for the patterns of movement required during bipedal walking. Comparative analysis of muscle attachment sites reveals how pelvic shape modifications altered the mechanical function of major locomotor muscles. The human pelvis provides attachment points that optimize muscle function for bipedal locomotion, while ape anatomy favors quadrupedal and arboreal movement patterns. These differences in muscle mechanics help explain how the same basic anatomical elements could be reorganized to support radically different locomotor behaviors. The magnitude of these anatomical differences initially puzzled evolutionary biologists. How could natural selection produce such dramatic structural modifications? What intermediate forms connected the ancestral ape-like condition with the derived human anatomy? The fossil record provided some insights into this evolutionary transformation, but the mechanisms responsible for such dramatic anatomical reorganization remained mysterious. Understanding these mechanisms required investigating the developmental processes that actually build pelvic anatomy during embryonic and fetal growth. If evolution modified the adult anatomy so dramatically, it must have done so by altering the genetic and developmental programs that construct the pelvis during individual development. This realization led researchers to examine embryonic specimens from humans and other primates, seeking to identify the specific developmental innovations that enabled our unique anatomy. The breakthrough in understanding human pelvic evolution required innovative research methods that could examine the actual processes of bone formation in developing embryos. This approach represented a significant departure from traditional paleontological methods, demanding access to precious embryonic specimens and sophisticated analytical techniques for studying three-dimensional anatomical development. Researchers utilized museum collections of embryonic specimens that had been preserved for decades in institutions around the world. These collections, assembled over many years for various research purposes, provided the raw material for comparative developmental studies. The specimens included human embryos at various gestational stages, as well as embryos from chimpanzees, gibbons, mice, and other mammals. The availability of such specimens made possible the first comprehensive comparison of pelvic development across primate species. The study of embryonic development required specialized preservation and imaging techniques. Embryonic tissues are delicate and require careful handling to maintain anatomical integrity. Researchers employed high-resolution microcomputed tomography scanning to create detailed three-dimensional images of developing skeletal elements. This non-destructive imaging technique allowed scientists to examine internal structures without damaging precious specimens, enabling multiple analyses of the same developmental stages. The temporal dimension of development presented particular challenges for research design. Unlike adult anatomy, which represents a stable end state, embryonic development is a dynamic process that unfolds over weeks and months. Researchers needed to sample multiple developmental time points to understand how anatomical structures change over time. This requirement demanded access to specimens representing the complete sequence of pelvic development, from initial formation through the onset of ossification. Comparative analysis across species required standardizing developmental stages between different mammals. Since gestation periods vary among primate species, researchers could not simply compare embryos of the same chronological age. Instead, they developed staging systems based on anatomical landmarks that allowed meaningful comparisons of equivalent developmental phases across species. This standardization proved crucial for identifying species-specific differences in developmental timing and patterning. The research focused particularly on the formation and early development of the ilium, the largest and most distinctive element of the pelvic girdle. In all mammals, the ilium begins as a rod of cartilage that forms during early embryonic development. This initial cartilaginous template serves as the foundation for subsequent bone formation and provides the basic structural framework that will eventually support adult locomotor function. Advanced microscopy techniques enabled researchers to examine the cellular processes responsible for cartilage and bone formation. They could observe the behavior of chondrocytes, the cells responsible for cartilage growth, and track the processes by which cartilage gradually transforms into bone tissue. This cellular-level analysis provided insights into the mechanisms by which developmental modifications could produce anatomical changes. The integration of developmental biology with evolutionary research required sophisticated analytical approaches. Researchers employed quantitative methods to measure anatomical features at different developmental stages, tracking how these measurements changed over time and how they differed between species. Statistical analysis helped identify significant developmental differences and distinguish meaningful biological variations from random developmental noise. Three-dimensional reconstruction techniques allowed researchers to visualize the complex spatial relationships involved in pelvic development. The pelvis is a three-dimensional structure with complex curvatures and spatial orientations that cannot be fully understood through two-dimensional analyses. Advanced imaging and reconstruction methods provided the spatial resolution necessary to identify subtle but important developmental modifications. The research also required careful attention to specimen quality and preservation. Embryonic tissues can suffer degradation over time, potentially obscuring important anatomical details. Researchers developed quality assessment protocols to ensure that only well-preserved specimens were included in analyses, maintaining the reliability and accuracy of their findings. Collaboration between institutions proved essential for assembling sufficient sample sizes for meaningful analysis. No single museum collection contained adequate specimens representing all the developmental stages and species necessary for comprehensive comparative analysis. International cooperation allowed researchers to access specimens from multiple collections, creating sample sizes adequate for rigorous statistical analysis. The methodological innovations developed for this research have applications beyond the specific question of pelvic evolution. The techniques for analyzing three-dimensional developmental changes over time can be applied to understanding the evolution of other anatomical systems. The integration of museum specimens with advanced imaging technologies provides a model for future research in comparative developmental biology. The initial phase of research focused on documenting the normal sequence of pelvic development in human embryos compared to other primates. Scientists examined specimens representing the earliest stages of skeletal formation, when the basic cartilaginous framework of the pelvis first becomes recognizable. This foundational work established the baseline developmental patterns necessary for identifying species-specific modifications. In all primates, pelvic development begins with the formation of three primary cartilaginous elements that will eventually become the major bones of the adult pelvis. The ilium, ischium and pubis initially develop as separate cartilaginous structures that gradually grow and fuse to create the integrated pelvic girdle. The ilium, being the largest of these elements and the most modified in human anatomy, became the primary focus of comparative analysis. During the 7th and 8th weeks of human gestation, the basic cartilaginous template of the ilium becomes clearly defined. At this stage, the structure appears as a relatively simple rod of cartilage extending from the developing hip joint. This early configuration is remarkably similar across primate species, suggesting that the initial phases of pelvic development follow conserved developmental pathways inherited from our common mammalian ancestry. The similarity of early developmental stages across species initially surprised researchers. Given the dramatic anatomical differences between adult human and ape pelvises, scientists had expected to find corresponding differences in early embryonic development. Instead, the early phases of development appeared nearly identical, suggesting that species-specific modifications occurred during later developmental stages, rather than at the initial formation of skeletal elements. Careful analysis of slightly later developmental stages began to reveal the first signs of species-specific differences. By the 9th and 10th weeks of human gestation, subtle but consistent differences became apparent in the orientation and growth patterns of the developing ilium. These early differences provided the first clues to the developmental mechanisms responsible for adult anatomical distinctions. The recognition of these early developmental differences required sophisticated analytical techniques. The modifications were subtle and could easily be overlooked without careful quantitative analysis. Researchers developed measurement protocols that could detect small but consistent differences in orientation, size and shape between human and ape embryonic specimens. Statistical analysis confirmed that these differences were biologically meaningful rather than random developmental variation. Documentation of developmental sequences required careful attention to the three-dimensional nature of anatomical growth. The developing ilium undergoes complex changes in orientation and curvature that cannot be fully captured through two-dimensional analysis. Researchers employed three-dimensional reconstruction techniques to track these changes over time, creating detailed developmental trajectories for each species examined. The temporal resolution of developmental analysis proved crucial for identifying critical phases of species-specific modification. Developmental changes occur rapidly during embryonic and fetal growth, with significant anatomical modifications occurring over periods of days or weeks. Researchers needed specimens representing closely spaced developmental time points to avoid missing important transitional phases. Comparative analysis revealed that while the initial formation of cartilaginous elements was similar across species, the subsequent growth and remodeling of these elements followed species-specific pathways. These differences in later developmental phases appeared to account for the dramatic anatomical differences observed in adult specimens. The identification of these developmental differences provided the foundation for understanding the evolutionary mechanisms responsible for human pelvic anatomy. The discovery of species-specific developmental differences raised new questions about the genetic and molecular mechanisms responsible for these modifications. If the basic developmental framework was conserved across primate species, what genetic changes enabled the species-specific modifications observed in later development? This question would drive the next phase of research, focusing on the molecular mechanisms controlling pelvic development. The documentation of normal developmental sequences also provided a framework for understanding pathological conditions affecting human pelvic development. Many congenital disorders affecting the pelvis involve disruptions of normal developmental processes. Understanding normal development provides the foundation for recognizing and potentially treating these developmental abnormalities. The most significant discovery emerged from detailed analysis of ileum development during the second and third months of human gestation. Researchers identified a remarkable developmental event that occurs consistently in human embryos but is absent in other primates. The iliac cartilage rod undergoes a dramatic 90-degree rotation from its original vertical orientation to a horizontal configuration perpendicular to the spine. This rotational event represents one of the most striking developmental differences ever documented between humans and other primates. In chimpanzees, gibbons and other apes, the iliac cartilage maintains its original vertical orientation throughout development, growing parallel to the vertebral column to create the tall, narrow pelvis characteristic of these species. Only in humans does this fundamental reorientation occur, setting the stage for the broad, bowl-shaped pelvis that enables upright locomotion. The timing of this rotational event proved crucial for understanding its significance. The rotation occurs during a critical phase of embryonic development when the basic body plan is being established and the skeletal framework is taking its final form. This early timing ensures that all subsequent growth and development of the pelvis proceeds along the modified trajectory established by the rotation. Detailed analysis revealed that the rotation is not a gradual process, but occurs as a discrete developmental event over a relatively short time period. Time series analysis of embryonic specimens showed that the ileum can complete its 90-degree rotation within a span of just a few weeks. This rapid transformation suggests that the rotation is controlled by specific developmental mechanisms rather than representing a gradual drift in growth orientation. The mechanical implications of this rotation are profound. By reorienting the ileum from a vertical to horizontal configuration, the rotation establishes the basic architectural framework that will enable bipedal locomotion. The horizontally oriented ileum provides the lateral stability necessary for single-leg stance during walking, while the vertical orientation retained by apes is optimized for quadrupedal and arboreal locomotion. Three-dimensional analysis of the rotation revealed its complex spatial nature. The ileum does not simply rotate around a single axis, but undergoes a complex three-dimensional transformation that involves rotation, translation, and changes in curvature. This complexity suggests sophisticated developmental control mechanisms that precisely orchestrate the spatial reorganization of the developing bone. The consistency of the rotation across human specimens examined was remarkable. Every human embryo analyzed showed evidence of this rotational event, with only minor variations in the precise timing and extent of the rotation. This consistency suggests that the rotation represents a fundamental feature of human development that is under tight genetic control and represents a derived characteristic of our lineage. Comparative analysis with other primates confirmed the uniqueness of this developmental event. Examination of chimpanzee, gorilla, and gibbon embryos revealed no evidence of iliac rotation. The ileum in these species maintains its original orientation throughout development, growing along pathways that create the tall, narrow pelvic configuration characteristic of adult apes. The discovery of iliac rotation provided the first clear developmental explanation for the fundamental anatomical differences between human and ape pelvises. This single developmental innovation could account for many of the distinctive features of human pelvic anatomy, including the broad iliac blades, the lateral orientation of muscle attachment sites, and the overall bowl-shaped configuration of the pelvic girdle. The evolutionary significance of this discovery cannot be overstated. The rotation represents a clear developmental synapomorphy, a derived characteristic that defines the human lineage and distinguishes it from all other primates. The identification of such a clear developmental marker provides powerful evidence for human evolutionary distinctiveness and offers insights into the timing and nature of human origins. However, the rotation alone could not account for all aspects of human pelvic anatomy. While the 90-degree rotation established the basic architectural framework for bipedal adaptation, additional developmental modifications were necessary to create the full suite of human anatomical characteristics. The identification of the rotation provided a foundation for investigating other developmental innovations that contributed to human anatomical evolution. The second major developmental innovation discovered by researchers involves the timing of bone formation in the human pelvis. While the 90-degree rotation of the iliac cartilage established the basic architectural framework for human pelvic anatomy, a second modification was necessary to create the full breadth and shape characteristic of our species. This second innovation involves a significant delay in the ossification or bone formation process that allows the cartilaginous pelvis to continue growing and expanding before hardening into bone. In typical mammalian development, cartilage serves as a temporary template that is gradually replaced by bone tissue through the process of endochondral ossification. Bone-forming cells called osteoblasts invade the cartilaginous template and begin depositing mineral matrix, gradually converting the flexible cartilage into rigid bone. The timing of this ossification process proves crucial for determining the final size and shape of skeletal elements. Comparative analysis of human and ape development revealed a striking difference in ossification timing. In chimpanzees and other apes, ossification of the ilium begins relatively early in fetal development, around 16 to 18 weeks of gestation. This early onset of bone formation limits the subsequent growth potential of the cartilaginous template, constraining the final size and shape of the adult bone. Human development follows a dramatically different timeline. Ossification of the human ilium is delayed until approximately 24 weeks of gestation, representing a delay of 6 to 8 weeks compared to other primates. This extended period of cartilaginous growth allows the human pelvis to continue expanding in width and depth before the onset of ossification fixes its final dimensions. The functional significance of this ossification delay becomes apparent when considering the biomechanical requirements of bipedal locomotion and human reproduction. The extended growth period allows the human pelvis to achieve the broad dimensions necessary for effective bipedal balance while maintaining adequate space within the birth canal for our species' characteristically large-brained infants. Detailed analysis of the ossification process revealed that the delay affects not only the timing, but also the pattern of bone formation. In humans, ossification proceeds more gradually and from multiple centers simultaneously, creating a more complex pattern of bone development. This modified ossification pattern contributes to the distinctive shape and internal architecture of human pelvic bones. The delay in ossification represents a clear example of heterochrony, an evolutionary mechanism in which changes in developmental timing produce anatomical modifications. Heterochrony has been recognized as a major mechanism of evolutionary change, particularly in mammalian evolution, where modifications of growth rates and timing have produced many adaptive anatomical innovations. The ossification delay also demonstrates the evolutionary flexibility of developmental mechanisms. Rather than requiring fundamental changes to the genetic programs controlling bone formation, evolution achieved significant anatomical modification through relatively simple alterations in developmental timing. This mechanism provides a pathway for rapid evolutionary change without disrupting basic developmental processes. Investigation of the molecular mechanisms controlling ossification timing revealed the involvement of multiple signaling pathways. The transition from cartilage to bone is controlled by complex interactions between growth factors, hormones, and transcription factors. Modifications in any of these regulatory systems could potentially alter ossification timing and produce evolutionary change. The discovery of delayed ossification provided important insights into the evolutionary sequence of human pelvic adaptations. The 90-degree rotation appears to have evolved first, establishing the basic architectural framework for bipedal adaptation. The ossification delay likely evolved later, fine-tuning the dimensions and proportions of the rotated pelvis to optimize both locomotor function and reproductive biology. This evolutionary sequence suggests a stepwise modification of human anatomy rather than a single dramatic transformation. Each developmental innovation built upon previous adaptations, gradually constructing the integrated system of anatomical features that enables human bipedalism. This incremental approach to evolutionary change helps explain how such complex anatomical systems could evolve through natural selection. The timing of these evolutionary innovations can be inferred from the fossil record and molecular phylogenetic analysis. The 90-degree rotation likely evolved early in hominin evolution, possibly coinciding with the initial commitment to terrestrial bipedalism around 5 to 7 million years ago. The ossification delay probably evolved later, possibly in conjunction with the evolution of increased brain size and the associated reproductive challenges around 1.5 to 2 million years ago. The identification of two major developmental innovations in human pelvic evolution raised fundamental questions about the genetic mechanisms responsible for these changes. How did evolution modify the genetic programs controlling cartilage orientation and ossification timing? What specific genes were involved in these developmental modifications? Understanding the molecular basis of human anatomical evolution requires examining the complex genetic networks that control skeletal development. Modern skeletal development involves hundreds of genes working in coordinated networks to control the formation, growth, and maturation of bone and cartilage tissues. These genetic networks have been conserved throughout vertebrate evolution, providing the basic toolkit for skeletal construction across all vertebrate species. However, subtle modifications in these genetic programs can produce significant anatomical changes, providing the raw material for evolutionary adaptation. Research into the genetic basis of pelvic development has identified several key regulatory genes that play crucial roles in controlling skeletal formation. The SOX9 gene functions as a master regulator of cartilage development, controlling the expression of numerous genes involved in cartilage formation and maintenance. Mutations in SOX9 cause severe skeletal malformations, demonstrating the critical importance of this gene in normal skeletal development. The RUNX2 gene serves as another crucial regulator, controlling the transition from cartilage to bone during the ossification process. RUNX2 activates the expression of genes necessary for bone formation while suppressing genes associated with cartilage maintenance. The timing and extent of RUNX2 expression therefore directly influence when and how cartilage is converted to bone during development. The PTH1R gene encoding the parathyroid hormone receptor, plays important roles in regulating both cartilage growth and ossification timing. This gene responds to hormonal signals that coordinate skeletal development with overall body growth and metabolic status. Modifications in PTH1R expression or function could potentially alter the timing of ossification and influence final skeletal proportions. Comparative genomic analysis between humans and other primates has identified numerous genetic differences that could potentially contribute to our distinctive skeletal anatomy. Many of these differences involve regulatory regions that control gene expression rather than changes to the protein coding sequences themselves. Such regulatory modifications can alter the timing, location, or intensity of gene expression without disrupting basic protein function. The complex nature of skeletal development means that anatomical evolution likely involves modifications in multiple genes working together rather than changes in single genes. The networks controlling skeletal development include extensive crosstalk between different signaling pathways, meaning that small changes in multiple genes could produce coordinated modifications in developmental outcomes. Experimental approaches using model organisms have provided insights into how genetic modifications can alter skeletal development. Studies in mice and other laboratory animals have demonstrated that targeted modifications of skeletal regulatory genes can produce anatomical changes reminiscent of those observed in human evolution. While these experimental systems cannot perfectly replicate human evolutionary history, they provide important insights into the mechanisms of skeletal evolution. The identification of candidate genes involved in human pelvic evolution opens new avenues for understanding human evolutionary history. Analysis of ancient DNA from Neanderthals, Denisovans, and other extinct human relatives could potentially reveal when specific genetic modifications occurred during human evolution. Such studies could provide direct evidence for the timing and sequence of genetic changes underlying human anatomical evolution. The genetic research also has important implications for understanding human health and disease. Many of the genes involved in normal pelvic development are also associated with congenital disorders affecting the pelvis and other skeletal elements. Understanding normal developmental mechanisms provides the foundation for developing new treatments for these conditions. The complexity of genetic networks controlling skeletal development suggests that human anatomical evolution involved sophisticated modifications of existing regulatory systems rather than the invention of entirely new genetic mechanisms. Evolution appears to have co-opted and modified ancient developmental programs, fine-tuning their operation to produce anatomically modern humans. Future research directions include more detailed analysis of gene regulatory networks, investigation of epigenetic mechanisms that could influence gene expression, and development of computational models that can predict the anatomical consequences of specific genetic modifications. These approaches promise to provide even deeper insights into the molecular mechanisms of human evolution. The anatomical innovations that enabled human bipedalism had consequences extending far beyond locomotion alone. Walking upright fundamentally altered human biology, behavior, and evolutionary trajectory in ways that continue to shape our species today. Understanding these broader implications provides crucial context for appreciating the full significance of the pelvic modifications that made bipedalism possible. The liberation of the hands represents perhaps the most obvious consequence of upright posture. When our ancestors no longer needed their hands for locomotion, these appendages became available for manipulation, tool use, and complex behavioral activities. This functional transformation enabled the development of sophisticated tool technologies that became central to human survival strategies and cultural evolution. Archaeological evidence documents a close temporal association between the evolution of bipedalism and the appearance of stone tool technologies in the archaeological record. The earliest recognized stone tools, dating to approximately 2.6 million years ago, appear in the same African contexts that preserve evidence of increasingly sophisticated bipedal adaptations. This temporal correlation suggests a functional relationship between upright posture and technological innovation. The biomechanical advantages of bipedal locomotion include significant improvements in energy efficiency for long-distance travel. Comparative studies of human and ape locomotion demonstrate that bipedal walking requires substantially less energy than quadrupedal locomotion at moderate speeds. This energy efficiency enabled early humans to travel longer distances while expending less metabolic energy, facilitating range expansion and resource exploitation strategies unavailable to other primates. Thermoregulatory benefits of upright posture also contributed to human adaptive success. The vertical body orientation associated with bipedalism reduces the surface area exposed to direct solar radiation during midday heat, while increasing the surface area available for convective heat loss. These thermoregulatory advantages proved particularly important as early humans expanded into more open savanna environments with limited shade cover. The elevated visual perspective provided by upright posture enhanced environmental awareness and predator detection capabilities. Standing upright allows humans to scan larger areas of their environment and detect potential threats or resources at greater distances. This improved situational awareness provided significant survival advantages in open environments where early detection of predators or prey could mean the difference between life and death. Bipedal locomotion also influenced human social behavior and communication systems. The upright posture facilitated visual communication across distances, enabling complex gestural communication systems that may have preceded the evolution of spoken language. The face-to-face orientation possible during bipedal locomotion enhanced opportunities for social interaction and the development of sophisticated social relationships. The reproductive implications of pelvic evolution extend beyond simple accommodation of large-brained infants. The modifications that enabled bipedal locomotion also influenced human reproductive physiology, including changes in gestation length, birth timing, and infant development patterns. These reproductive modifications had cascading effects on human social organization, parental care strategies, and life history evolution. The evolution of increased brain size in human ancestors created additional selective pressures that influenced pelvic evolution. Larger brains required larger birth canals, but the biomechanical requirements of bipedal locomotion constrained pelvic dimensions. The evolutionary solution involved the complex balancing act reflected in modern human pelvic anatomy, which represents a compromise between locomotor efficiency and reproductive function. Dietary implications of bipedal evolution include changes in foraging strategies and food processing techniques. Upright posture enabled more efficient long-distance travel for resource acquisition, while freed hands allowed more sophisticated food processing and tool use. These capabilities expanded the range of food resources available to early humans and contributed to dietary diversification. The cultural implications of bipedalism are perhaps the most profound and far-reaching. The combination of freed hands, enhanced mobility, and improved environmental awareness created the foundation for the complex technological and cultural systems that define modern human societies. The capacity for tool manufacture and use, long-distance trade, artistic expression, and symbolic communication all trace their origins to the anatomical innovations that enabled upright walking. Modern human anatomy and physiology continue to reflect our bipedal heritage. Many contemporary health issues, including back problems, hip disorders and complications during childbirth, can be traced to the evolutionary compromises involved in adapting to upright posture. Understanding these evolutionary origins provides important insights into the nature of human health and the ongoing challenges of living in bodies designed for environments very different from our modern world. The human spine exemplifies the evolutionary compromises inherent in bipedal adaptation. Our distinctive S-shaped spinal curvature, while essential for upright posture, creates mechanical stress points that make us vulnerable to disc herniation, vertebral compression, and chronic back pain. The lower lumbar region bears the greatest load during upright posture and consequently experiences the highest rates of degenerative changes throughout human lifespans. Obstetric complications in humans occur at rates far higher than in other primates, reflecting the tight evolutionary constraints on pelvic dimensions. The human birth canal represents a carefully balanced compromise between the biomechanical requirements of efficient bipedal locomotion and the anatomical needs of safe childbirth. This balance becomes increasingly precarious as infant head sizes continue to approach the limits of what the birth canal can accommodate. Hip disorders, including developmental dysplasia and early arthritis, also reflect our bipedal heritage. The human hip joint operates under mechanical loads and stress patterns that differ fundamentally from those experienced by quadrupedal mammals. The weight-bearing demands of upright posture, combined with the complex three-dimensional movements required during human locomotion, create unique challenges for hip joint function and longevity. The revolutionary discoveries about human pelvic evolution were made possible by advances in imaging technology, computational analysis, and interdisciplinary collaboration that continue to open new frontiers for understanding human evolutionary history. These technological capabilities promise to reveal additional secrets about our evolutionary past while providing new tools for addressing contemporary health challenges. High-resolution microcomputed tomography has transformed the study of fossil and embryonic specimens by enabling non-destructive internal visualization of delicate structures. This technology allows researchers to examine the internal architecture of precious fossil specimens without risk of damage, while providing unprecedented detail about developmental processes in embryonic material. Continued improvements in imaging resolution and processing speed will enable even more sophisticated analyses of evolutionary changes. Three-dimensional reconstruction and modeling technologies have become essential tools for understanding complex anatomical relationships and developmental processes. These techniques allow researchers to visualize and quantify changes in three-dimensional space over time, providing insights into the spatial dynamics of evolutionary transformation. Virtual reality and augmented reality technologies are beginning to provide new ways of visualizing and interacting with complex anatomical data. Advances in ancient DNA extraction and analysis techniques are opening new possibilities for understanding the genetic basis of human evolutionary changes. While DNA preservation is limited in most fossil contexts, successful extraction of genetic material from Neanderthal and Denisovan remains demonstrates the potential for directly examining the genetic changes underlying human anatomical evolution. Continued improvements in ancient DNA techniques may eventually allow analysis of much older specimens. Computational modeling approaches are becoming increasingly sophisticated in their ability to predict the functional consequences of anatomical modifications. Finite element analysis, biomechanical modeling, and evolutionary simulations provide powerful tools for testing hypotheses about the adaptive significance of anatomical changes. These approaches allow researchers to explore evolutionary scenarios that cannot be directly observed in the fossil record. CRISPR and other gene editing technologies offer new possibilities for experimentally testing hypotheses about the genetic basis of human anatomical evolution. While ethical considerations limit direct experimentation on human development, studies in model organisms can provide important insights into the consequences of specific genetic modifications. These experimental approaches complement observational studies by providing direct evidence for cause and effect relationships. Machine learning and artificial intelligence applications are beginning to revolutionize the analysis of complex anatomical data sets. These computational approaches can identify subtle patterns in anatomical variation that might be missed by traditional analytical methods. Machine learning algorithms can also integrate information from multiple sources, including genetic, developmental, and fossil data, to provide more comprehensive understanding of evolutionary processes. The integration of developmental biology with paleontology continues to provide new insights into evolutionary mechanisms. This interdisciplinary approach, sometimes called evolutionary developmental biology or EVO-DEVO, examines how changes in developmental processes produce evolutionary change. The success of this approach in understanding human pelvic evolution suggests its potential for illuminating other aspects of human evolutionary history. International collaboration and data-sharing initiatives are accelerating the pace of discovery by making specimens and datasets available to researchers worldwide. Digital repositories of anatomical data, genetic sequences, and imaging datasets enable collaborative research projects that would be impossible for individual laboratories. These collaborative approaches are particularly important for studies requiring large sample sizes or rare specimens. The development of new analytical techniques for studying fossilization processes is improving our understanding of what information can be reliably extracted from fossil specimens. Taphonomic studies examine how biological materials are preserved in the fossil record, helping researchers distinguish between genuine biological features and artifacts of preservation. This understanding is crucial for correctly interpreting fossil evidence of developmental processes. Advances in tissue engineering and regenerative medicine are providing new insights into the developmental processes that build skeletal structures. These biomedical applications often require detailed understanding of normal developmental mechanisms, creating synergies between evolutionary research and medical applications. The knowledge gained from studying human pelvic evolution may contribute to developing new treatments for developmental disorders and injuries. Despite the remarkable progress in understanding human pelvic evolution, numerous questions remain unanswered, providing exciting opportunities for future research. These outstanding questions span multiple scales of analysis, from molecular mechanisms to ecosystem-level interactions, and require continued interdisciplinary collaboration to address adequately. The precise timing of the evolutionary innovations discovered in human pelvic development remains uncertain. While researchers can infer approximate timing from phylogenetic analysis and fossil evidence, direct evidence for when these developmental modifications first appeared would provide crucial insights into human evolutionary history. Future discoveries of well-preserved fossil specimens or successful extraction of ancient DNA could provide more precise temporal frameworks. The relationship between pelvic evolution and other aspects of human anatomical modification requires further investigation. The evolution of bipedalism involved coordinated changes throughout the locomotor system, including modifications to the vertebral column, lower limb bones, and associated musculature. Understanding how these various anatomical changes were integrated during evolution will provide a more complete picture of bipedal adaptation. The genetic mechanisms controlling the 90-degree rotation of the iliac cartilage remain largely mysterious. While researchers have identified candidate genes involved in skeletal development, the specific molecular mechanisms responsible for this dramatic developmental modification have not been fully elucidated. Experimental approaches using model organisms may provide insights into the genetic networks controlling spatial orientation during skeletal development. The evolutionary forces that drove the initial commitment to bipedal locomotion continue to be debated among researchers. Multiple hypotheses have been proposed, including thermoregulatory advantages, foraging efficiency, predator avoidance, and social display functions. Testing these alternatives requires integrating evidence from multiple sources, including biomechanical analysis, ecological reconstruction, and comparative studies of primate behavior. The relationship between brain size evolution and pelvic modifications represents another area requiring further research. The evolutionary expansion of human brain size created selective pressures for larger birth canals, potentially influencing the timing and extent of pelvic modifications. Understanding this relationship requires detailed analysis of both neurological and skeletal evolution throughout human evolutionary history. The role of developmental plasticity in human pelvic evolution deserves additional attention. Modern human populations show considerable variation in pelvic anatomy, some of which reflects responses to different environmental conditions during development. Understanding the extent and mechanisms of developmental plasticity could provide insights into how evolutionary modifications became established in human populations. The comparative analysis of pelvic development could be extended to additional primate species and other mammalian groups. While current research has focused primarily on great apes as the closest relatives to humans, analysis of more distantly related primates could provide insights into the evolutionary origins of the developmental mechanisms involved in pelvic formation. Studies of other bipedal mammals could reveal whether similar developmental modifications evolved independently in other lineages. The application of new technological approaches continues to open additional research possibilities. Improved imaging techniques, more sophisticated computational models, and advances in ancient DNA analysis will provide new tools for addressing outstanding questions about human evolution. The integration of these diverse approaches promises to provide increasingly detailed understanding of evolutionary processes. The medical implications of evolutionary research on human pelvic anatomy require further exploration. Understanding the evolutionary origins of anatomical features associated with common health problems could inform the development of new diagnostic and treatment approaches. This translational research represents an important opportunity to apply evolutionary insights to contemporary health challenges. The educational and public outreach implications of this research deserve attention as well. The story of human pelvic evolution provides a compelling example of how scientific research can reveal fundamental aspects of human nature and evolutionary history. Communicating these discoveries to broader audiences can enhance public understanding of evolutionary science and its relevance to human health and welfare. The scientific journey to understand how humans became upright represents one of the most successful interdisciplinary research programs in modern biology. Through the integration of paleontology, developmental biology, genetics and biomechanics, researchers have revealed the specific mechanisms that enabled our species' most distinctive anatomical adaptation. These discoveries fundamentally change our understanding of human evolutionary history and provide new frameworks for addressing contemporary challenges in health and medicine. The identification of two major developmental innovations in human pelvic evolution demonstrates the power of studying evolutionary processes at the level of individual development. The 90-degree rotation of the iliac cartilage and the delay in ossification timing represent discrete identifiable changes that can be traced through the fossil record and analyzed using modern molecular techniques. This mechanistic understanding of evolutionary change provides a model for investigating other aspects of human anatomical evolution. The research reveals evolution as a process of creative problem-solving, finding innovative solutions to complex engineering challenges. The human pelvis represents an elegant compromise between the competing demands of efficient bipedal locomotion and safe reproduction of large-brained offspring. This evolutionary solution required precise modifications of developmental timing and spatial organization, demonstrating the sophisticated ways in which natural selection can modify biological systems. The broader implications of bipedal evolution for human biology and behavior underscore the profound consequences of anatomical change. The liberation of the hands, improvements in locomotor efficiency, and enhanced environmental awareness enabled by upright posture created the foundation for the technological and cultural achievements that define our species. Understanding these connections between anatomy and behavior provides crucial insights into what makes us uniquely human. The ongoing relevance of evolutionary research to contemporary human health demonstrates the practical importance of understanding our evolutionary heritage. Many modern health challenges, from back problems to childbirth complications, reflect the evolutionary compromises involved in adapting to bipedal locomotion. This understanding provides new perspectives on prevention, diagnosis, and treatment of these conditions. The technological advances that enable these discoveries continue to open new frontiers for evolutionary research. High-resolution imaging, computational modeling, and genetic analysis techniques provide unprecedented capabilities for examining evolutionary processes at multiple scales of organization. The integration of these diverse approaches promises continued breakthroughs in understanding human evolutionary history. The collaborative nature of this research exemplifies the benefits of interdisciplinary scientific investigation. The combination of expertise from multiple fields, access to diverse specimen collections, and application of varied analytical techniques created synergies that would have been impossible within traditional disciplinary boundaries. This collaborative model provides a template for addressing other complex scientific questions. The story of human pelvic evolution also highlights the importance of museum collections and long-term specimen preservation for scientific research. The embryonic specimens that enabled these discoveries were collected and preserved over many decades by institutions worldwide. This research demonstrates the continuing scientific value of museum collections and the importance of supporting these repositories for future research. The educational value of this research extends beyond its specific scientific contributions. The story of how humans became upright provides a compelling example of the scientific process in action, demonstrating how careful observation, innovative methodology, and persistent investigation can reveal fundamental truths about the natural world. This narrative can inspire future generations of scientists and enhance public appreciation for scientific research. Looking toward the future, The framework established by this research provides a foundation for investigating other aspects of human evolutionary history. The same approaches that revealed the mechanisms of pelvic evolution can be applied to understanding the evolution of other distinctive human characteristics, from brain expansion to language capabilities to extended lifespans. The ultimate significance of understanding how humans became upright extends beyond scientific curiosity to fundamental questions about human nature and our place in the natural world. The anatomical innovations that enabled our ancestors to stand tall initiated a cascade of evolutionary changes that ultimately led to the emergence of modern human civilization. Every step we take connects us to this deep evolutionary heritage and reminds us of the remarkable journey that brought us to where we stand today. The research reveals that becoming upright was not a single evolutionary event, but a complex process involving multiple anatomical innovations occurring over millions of years. This incremental approach to major evolutionary change provides important insights into how complex biological systems evolve and how seemingly impossible transformations can occur through gradual modification of existing structures. The precision required for the developmental modifications that enabled bipedalism underscores the remarkable capabilities of evolutionary processes. The coordination required between spatial reorientation and temporal delays in development demonstrates that evolution can produce sophisticated solutions to complex problems through the accumulation of beneficial mutations over time. As we continue to walk upright through our daily lives, we carry within our bodies the legacy of these ancient evolutionary innovations. The bowl-shaped pelvis that enables our upright posture connects us directly to our earliest human ancestors and reminds us of the long journey that brought our species from the trees to our current position as the dominant terrestrial bipeds on Earth. The story of human bipedalism ultimately reveals evolution as a creative process that can produce remarkable innovations through the modification of existing biological systems. The transformation of a quadrupedal primate into an efficient biped required fundamental reorganization of anatomy, physiology, and behavior, yet evolution accomplished this transformation through the accumulation of relatively simple developmental modifications. Understanding how we became upright provides crucial context for appreciating the full scope of human evolutionary achievement. The anatomical foundation established by bipedal adaptations enabled all the subsequent innovations that characterize modern human societies, from complex technology to symbolic communication to global civilization. In learning how we learn to walk, we gain essential insights into what makes us fundamentally human.